Welcome to our lecture on housefly control, a crucial topic in medical entomology. Imagine enjoying a summer picnic, only to have a fly land on your food after crawling on decaying waste. Houseflies are not just annoying. They are essentially flying garbage trucks that transport dangerous bacteria directly to us. Today, we will explore how to scientifically manage their populations and protect public health. To begin, we must understand why houseflies are so dangerous. Houseflies act as mechanical vectors. This means they do not bite or inject pathogens into our blood like mosquitoes do. Instead, they pick up disease-causing organisms from filth using their hairy legs and spongy mouthparts and physically carry them to our food and living spaces. Because they reproduce at an astonishingly rapid rate, simply swatting them one by one is a losing battle. We need a deeper understanding of their biology to stop them. Now that we know how they carry pathogens, let us look at the specific damage they cause. Houseflies are responsible for transmitting severe gastrointestinal diseases, such as cholera, typhoid fever, dysentery, and salmonellosis. But the threat extends beyond human households. In agricultural settings, massive fly populations severely stress livestock, which directly reduces milk and meat production. Furthermore, in hospital environments, flies can land on open wounds and cause secondary infections, a dangerous condition known as myiasis. Without strict control, densely populated areas would face devastating outbreaks. Understanding these severe impacts brings us to our primary defense strategy, integrated vector management, or IVM. In medical entomology, housefly control is not about eradicating every single fly on Earth. That is impossible. Instead, the goal is to systematically manage the Musca domestica population, keeping it below a threshold where it can cause public health emergencies. IVM achieves this by combining environmental, mechanical, biological, and chemical strategies. Crucially, these measures must target both the immature larval stages, known as maggots, and the highly mobile adult flying stages to break the life cycle. Let us visualize this multi-layered approach using the IVM pyramid. At the wide base of the pyramid, we have cultural control, which forms the foundation through strict sanitation and waste management to remove breeding grounds. Moving up, the next layer is physical and mechanical control, utilizing screens and traps to create physical barriers. Above that is biological control, where we introduce natural predators like parasitoid wasps. Finally, at the very top, we have chemical control, using insecticides. Notice that chemicals are the smallest part of the pyramid, representing an intervention of last resort when other methods are not enough. As we saw at the top of the pyramid, chemical control is our last resort. When sanitation and mechanical traps are not enough, public health officials turn to chemical insecticides to knock down adult housefly populations quickly. However, before spraying chemicals everywhere, we must scientifically prove that the chemical actually works. We do this by comparing a treated population of flies against an untreated control population. This comparison is absolutely vital because it accounts for natural deaths that would have happened anyway, ensuring we measure only the mortality caused by the insecticide itself. To mathematically calculate this true effectiveness, we use Abbott's formula. Conceptually, this formula isolates the deaths caused specifically by the chemical by finding the difference between the control and treated survivors. In this equation, E represents the percentage efficacy of the insecticide treatment. C stands for the number of living insects in the untreated control group after a specific period. T represents the number of living insects in the chemically treated group after that exact same period. If the value of T decreases, meaning fewer flies survive the spray, the numerator becomes larger, and the overall efficacy, E, increases toward 100%. Before we move on, let us see this with a concrete example. Imagine a dairy farm experiencing a massive housefly infestation. The farmer applies a new chemical spray to one barn, leaving an identical barn untreated as a control. After 24 hours, we are given that the untreated control barn has 500 live flies remaining, so C equals 500. The treated barn has only 50 live flies remaining, so T equals 50. We are asked to calculate the exact efficacy percentage. We choose Abbott's formula to account for natural environmental factors. Here, we substitute our known values of 500 and 50 into the equation, setting up the numerator to find the exact number of flies killed by the intervention. 500 minus 50 gives us 450 dead flies. Continuing with our calculation, we take the 450 eliminated flies and divide them by the baseline control population of 500. This division gives us the raw decimal proportion, which is 0.90. Finally, multiplying this decimal by 100 shifts the decimal point 2 places to the right, converting it into a recognizable percentage. Our final answer is 90%. Physically, this means the insecticide successfully eliminated 90% of the target housefly population, proving it is a highly effective tool for this farm. Now that we have used this formula to measure control, let us understand why control must be implemented so rapidly in the first place. If a single female housefly finds a suitable breeding ground, she lays a batch of eggs. Biological populations do not grow by simply adding a fixed number of new flies each day. Instead, they multiply. The offspring reproduce, and their offspring reproduce, leading to what we call exponential growth. Because of this compounding effect, even a small initial population can explode into a massive infestation in a very short time. Delaying sanitation or control measures is mathematically disastrous. To project exactly how fast this explosion happens, we use the standard exponential growth formula. Conceptually, this equation models continuous compounding growth in population ecology. Let us break down the symbols. N sub t is the final population size after a certain time. N sub 0 is our initial starting population. The variable r is the intrinsic daily growth rate of the species, and t is the time period in days. Finally, E is Euler's number, a mathematical constant, approximately equal to 2.718, which serves as the base for continuous growth. If the growth rate r, or the time t, increases, the exponent grows, causing the final population to skyrocket. Let us apply this formula to see the mathematical threat of a delayed response. We are given a scenario where a garbage can breeds an initial population of 100 surviving adult flies, so N sub 0 is 100. Under warm summer conditions, the intrinsic growth rate is 0.2 per day, giving us our R value. We are asked to find the population size in exactly 10 days, so T equals 10. We choose the exponential growth formula because biological populations multiply continuously. By substituting our initial population, growth rate, and time into the equation, we set up the exponent to find the total accumulated growth factor over this 10-day period. Our equation is now ready to be evaluated. Now, let us solve the equation step by step. First, we multiply the daily growth rate of 0.2 by the 10 days in the exponent, which simplifies down to exactly 2. This means the population will grow by a factor of e squared. Evaluating e to the power of 2 gives us approximately 7.389, which is our total growth multiplier. Multiplying this multiplier by our initial 100 flies yields 738.9. Since we cannot have a fraction of a fly in a real-world biological context, we round this to a final answer of 739 living adult flies. This explosive increase perfectly illustrates why early sanitation is critical. We have covered a lot of ground today, so let us review our key takeaways. First, remember that houseflies are dangerous mechanical vectors that transmit deadly pathogens without biting. Second, because their populations grow exponentially, early intervention through strict sanitation is absolutely critical to prevent massive outbreaks. Third, we cannot rely on a single method. We must use integrated vector management, starting with sanitation and ending with chemicals as a last resort. Finally, when we do use chemicals, Abbott's formula allows us to scientifically measure their true efficacy. You now have a solid understanding of both the biology and the mathematics behind housefly control. Great job!